Welcome to Biology, the scientific study of life, the genetics of viruses and bacteria. Microbial model systems. This photo in this figure shows a remarkable event, the attack of a bacterial cell by numerous structures that resemble miniature lollipops. These structures, a type of virus called T4 bacteriophage, are seen infecting by bacterium Escherichia coli in this colorized SAM by injecting its DNA into the cell. The virus sets in motion a genetic takeover of the bacterium. Molecular biology was born in the laboratories of microbiologists studying viruses and bacteria. Microscopes such as E. coli and its viruses are called model systems because of their frequent use by researchers in studies that reveal broad biological principles. Experiments with viruses and bacteria provided most of the evidence that genes are made of DNA, and they were critical in working out the molecular mechanisms of the fundamental processes of DNA replication, transcription, and translation. Beyond their value as model systems, viruses and bacteria have unique genetic mechanisms that are interesting in their own right. These specialized mechanisms have important applications for understanding how viruses and bacteria cause disease. In addition, techniques enabling scientists to manipulate genes and transfer them from one organism to another have emerged from the study of microbes. These techniques are having an important impact on both basic research and biotechnology. In this chapter, we explore the genetics of viruses and bacteria. Recall that bacteria are prokaryotes with cells much smaller and more simply organized than those of eukaryotes, such as plants and animals. Viruses are smaller and simpler still, lacking the structures and metabolic machinery found in cells. Most viruses are little more than genes packaged in protein codes. We will begin with the structure of this simplest of all genetic systems and their role as disease-causing agents or pathogens. Then we will discuss the genetics of bacteria and regulation of their gene expressions. A virus has a genome but can reproduce only within a host cell. Scientists were able to detect viruses indirectly long before they were actually able to see them. The story of how viruses were discovered begins near the end of the 19th century. The discovery of viruses, scientific inquiry. Tobacco mosaic disease stunts the growth of tobacco plants and gives their leaves a mottled or mosaic coloration. In 1883, Adolf Meyer, a German scientist, discovered that he could transmit the disease from plant to plant by rubbing sap extracted from diseased leaves into healthy plants. After an unsuccessful search for an infectious microbe in the sap, Meyer concluded that the disease was caused by unusual small bacteria that could not be seen with the microscope. This hypothesis was tested a decade later by Dmitry Ivanovsky, a Russian who passed sap from infected tobacco leaves through a filter designed to remove bacteria. After filtering, the sap still produced mosaic disease. Ivanovsky clung to the hypothesis that bacteria caused tobacco mosaic disease. Perhaps he reasoned the bacteria were so small that they passed through the filter or made a filterable toxin that caused the disease. This later possibility was ruled out when the Dutch botanist Matronus Beijerinck discovered that the infectious agent in the filtered sap could reproduce he rubbed plants with the filtered sap, and after these plants developed mosaic disease, he was their sap to infect more plants, continuing this process through a series of infections. The pathogens must have been reproducing, for its ability to cause disease was undiluted after several transfers from plant to plants. In fact, the pathogen reproduced only within the host it infected. Unlike bacteria, the mysterious agent of mosaic disease could not be cultivated on nutrients media in test tube or petri dishes. Picturing imagined a reproducing partial, much smaller and simpler than bacteria. His suspicions were confirmed in 1935 when the American scientist Winrow Stanley crystallized the infectious partial, now known as tobacco mosaic virus, TMV, subsequently TMV, and many other viruses were actually seen with the help of the electron microscope. Notice that. Infection by tobacco mosaic viruses. Notice that this photo. A healthy uninfected tobacco and compared with a leaf experimentally infected. Notice that. Back to comparing the size of a virus, bacterium, and an animal cell. Only a portion of a typical animal cell is shown. Its diameter is about 10 times greater than the length of E. coli. Notice here virus and bacterium, animal cell. Structure of viruses. The tiniest viruses are only 20 nanometer in diameter, smaller than a ribosome. Millions could easily fit on a pinhead. Even the largest viruses are barely visible in the light microscope. Stanley's discovered that some viruses could be crystallized was existing and puzzling news. Not even the simplest of cells can aggregate into regular crystals. But if viruses are not cells, then what are they? They are infectious partials consisting of nucleic acid enclosed in a protein coat, and in some cases a membranous envelope. Let's examine the structure of viruses more closely and then how they reproduce. Viral genome. We usually think of genes as being made of a double-stranded DNA, the conventional double helix, but many viruses defy this convention. Their genomes may consist of double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA, depending on the kind of virus. A virus is called a DNA virus or an RNA virus, according to the kind of nucleic acid that makes up its genome. In either case, the genome is usually organized as a single linear or circular molecule of nucleic acid. The smallest viruses have only 4 genes, while the largest have several hundred.